All right, hello, I'm Katherine Geiersbach, and I'm an associate professor in the Department of Laboratory Medicine and Pathology at Mayo Clinic in Rochester. And I've been there for about 6 years. I hope you guys are all having a great meeting. It's a memorable meeting for me for a variety of reasons, but one of which I will not forget this experience. I went to a beautiful butterfly garden about 30 miles west of here on my way in because I love outdoors, and I got unfortunately into a patch of chiggers, and I don't know if anybody else from here is from the Midwest, but it's a very unpleasant experience having chigger bites. So I'm recovering. But I wanted to also thank Thermo for the opportunity to speak about our lab's experience here with SNP array testing on melanocytic tumors. Mayo Clinic has a very large dermatopathology consultation practice, and as part of that, they run ancillary testing to help with the diagnosis of basically borderline challenging melanocytic tumors. And I'm going to provide some background here. But first, a disclaimer that you can read. And so as background on the gross and microscopic diagnosis of melanoma, grossly, I think many of you are familiar with this and self-screening for melanoma. So the ABCDE method of grossly diagnosing melanoma evolved, mid-1980s, and then was introduced into the clinic as a standard way to basically diagnose possible melanoma. And of course, the standard being histologic examination, and what I'm showing here is on the top a clear-cut benign nevus, and on the bottom is invasive melanoma, and I think we're all aware that invasive melanoma can be deadly once it's no longer localized. Very important to diagnose melanoma accurately. And so the traditional criteria for diagnosing melanoma are subjective, and they really vary with the experience of either the dermatologist or the dermatopathologist. And as you can see here in the table, I'm showing that for the ABCDE clinical criteria, if we use over two criteria or two or more criteria, quite good sensitivity, almost 90% there, but not as good specificity. Using 3 or more of the ABCDE criteria, the sensitivity does go down to 66%, but improves the specificity for clinically diagnosing melanoma based on a reference standard of basically expert pathology consensus diagnosis. And then for histopathology, again, the sensitivity is 69% for an individual pathologist, but quite highly specific for melanoma. And it turns out that actually the histologic diagnosis of melanoma is quite challenging, and I've shown here some quotes that I pulled from some recent papers, which the top quote along the bottom of my slide shows there are significant discordance rates using routine examination between different pathologists. And then the second quote I have here highlights that there's so the distinction between benign and malignant melanocytic neoplasms remains one of the most formidable challenges in dermatopathology. And so basically, there are many cases in which histopathologic diagnosis is sufficient, and so sending to a consultation service that's a very high volume practice is sort of the first step if there is a challenging histology, and for the most part, that is quite sufficient, maybe some ancillary immunostains, but then following up on that in a good fraction of cases, the genetics becomes very useful for determining whether tumor is potentially malignant. So karyotype is sort of, actually was interesting in the 1980s. They knew that terminal 9p deletions were sort of a hallmark of many melanomas. And I think one of the authors on the paper was Uta Franke. Then FISH, of course, remains in use now, but has become supplanted, I think, more and more now by SNP array. The previous technology, which was used as CGH, comparative genomic hybridization, is now more and more SNP array. And so I'm going to talk about the lab's experience with clinical testing on SNP array using a Thermo Fisher platform. And then TERT promoter mutation has emerged as another method that could be helpful for distinguishing between malignant and benign, and gene expression profiling, such as the Castle Biosciences test that was previously owned by Myriad, which basically the expression of a group of genes is combined into an individual score, and you can see two distributions. On the bottom right image, where you can see in the darker gray the tumors are showing one distribution versus the bottom on the left. So in unequivocal benign nevi or malignant melanomas, it turns out that the performance of these various technologies is quite good. And so this is the basis for most of the clinical validation studies, because we know ground truth. Ground truth can be expert pathology consensus, or it can be disease follow-up, or both, as I've shown here. And so really the sensitivity ranges from high 80s to low 90s with excellent specificity, particularly for the publications using SNP array. But then the question really becomes whether the intended use is really in these tumors that are equivocal, or they're borderline, they're ambiguous. In other words, we cannot easily distinguish benign versus malignant. And the tumor has, of course, been biopsied, usually excised in toto. So it's basically removed from the body, therefore altering the clinical course. We don't know that if we had left the tumor in place, would it have progressed to metastatic disease. And so it's hard to really have a ground truth with some of these tumors. And but we do have some data from clinical studies on FISH and gene expression profiling and SNP array, showing that the sensitivity for basically comparing to integrated diagnosis. So that means taking all of the data, including usually SNP array or whatever the ancillary method is, and combining the data. We have sensitivity between 80% and mid 90%. So basically showing that the performance, while it is of a little bit lower sensitivity, holds true, at least as far as we can tell. I also wanted to highlight that clinical practice guidelines support the use of array testing, and as you may have seen earlier in Cynthia's talk on a systematic review of melanomas, the American Society of Dermatopathology has established criteria for many clinical scenarios, and array is a preferred method. And gets the higher scores, although gene expression profiling is also an emerging method that is increasingly used in this setting for determining benign versus malignant, and it's accepted for all except for basically the non-ambiguous benign or non-ambiguous malignant tumors. And again, the NCCN guidelines sort of classify array testing or genome-wide copy number profiling, which they list CGH and SNP array as a preferred method, highlighting its superior sensitivity and specificity because of the genome-wide nature of the ability to detect any copy number alteration. And then gene expression profiling is, again, They do highlight that there is inconsistently identified gene signatures between different studies. Again, I won't belabor this, but I wanted to bring out that the differences in copy number alterations along the x-axis, you can see along the top the malignant tumors, along the bottom are the benign tumors, and then everything else is the intermediate. And there's quite a bit of overlap in the intermediate tumors in terms of, in this large systematic review, there was a large amount of overlap in terms of some intermediate tumors having no copy number alterations and some having multiple copy number alterations in the range of the malignant tumors. So many of you may be familiar with SNP array and the molecular inversion probe technology that Thermo is using for its OncoScan platform, and it's shown here. It's basically, the idea is there is a probe-based technology that hybridizes and circularizes once it's hybridized, and the non-circular nucleic acids are digested away, and only the circular template remains for amplification and hybridization to the array, and seems to work quite well in highly degraded DNA or poor quality DNA, such as you get from paraffin-embedded tissue. And this is a busy chart, so what I really wanted to just emphasize is I'm going to be talking about the lab experience in two different cohorts. One is sort of the non-ambiguous, clear-cut sort of clinical validation cases where we know ground truth, we know the samples are either from a clinically benign tumor or a clinically malignant tumor, and then the others are unknowns. And I just wanted to also show our results. This is unpublished data that we had, and it's difficult for me to see this, but 8% of our benign tumors were abnormal. Thank you. It's tiny. 8% abnormal for the benigns and 100% abnormal for malignant tumors. Whereas on the right, you can see that the benign tumors most of them had less than, or all of them had less than 3 copy number abnormalities. And again, these were informed in part by the SNP array result. So this is not independent data, but basically the ambiguous tumors shown over on the right side in the light blue section ranged in the number of abnormalities, and we subdivided them into likely benign or likely malignant or still ambiguous. But the SNP array results could help inform the final diagnostic compression. And this is basically all of the data just shown in a genome-wide plot. And you can see as we progress from those pathology consultation unknown cases that were almost certainly clinically benign through the ambiguous to the malignant tumors, increasingly the tumors begin to resemble in their profile known malignant tumors. And then the TCGA data along the bottom showing a similar distribution of copy number alterations. And again, as Cynthia mentioned, the duplication on 11p that you can see along the top, I guess I'll use a pointer. This 11p duplication is more likely to be associated, it favors a benign tumor over malignant, but otherwise most of the copy number alterations are seen primarily in malignant tumors. So just boiling it down to some recommended best practices that we came across in our experience that we have developed. Our method includes masking, of course, known benign alterations. Using disease-specific gene lists, this becomes important for identifying breakpoints in kinase genes. It's a common recurrent abnormality in spitzoid tumors. And using our validated reporting thresholds, and then in our interpretation, a big piece of this is evaluating the overall acceptability of the data, because the results tend to be noisier by far compared to other tumor types, and we're not entirely sure why yet. It may have a little bit to do with the very limited amount of DNA we get from many of these small skin biopsy samples. And we obviously have to consider the abnormalities in the context of the patient, the disease. And then finally, these results are considered ancillary data and integrated with the rest of the data on the patient. So I'm just going to show a few quick cases as just illustrating some of the challenges we come across. So here's just an example of an original attempt and then a repeat attempt that shows cleaner data. Basically what we did is we increased the number of slides from our standard 10 unstained sections to 15. We got more DNA, and in this case, it cleaned up our data very nicely. Still, again, most of our melanoma samples yield a little bit noisier data, but the copy number alterations we're looking for are by and large, very large ones, and so we can still interpret the results. And then I'm just showing, again, the very small duplication on 2P over here, and then zooming in, you can see much cleaner results on the blue, which is a repeat. And then there's a 9Q deletion, and again, in the blue is our repeat attempt. It just shows cleaner data, better distinguishing our breakpoints. And so basically what we have is a 2P gain and a 9Q loss using our gene lists. We don't really have any genes of interest here. Overall less than 3 abnormalities, which is sort of that heuristic threshold that we've been using for approximately distinguishing between benign and malignant. And so this is essentially an atypical lesion. Typically, so the samples that are sent in for array, if they don't show anything that's consistent with malignant, if in other words it's not helping with the diagnosis of malignancy, it usually stays as an atypical, or in other words, borderline lesion. And then I'm just showing a distribution here, just straight across on the left. What is the typical DNA yield for 10 sections on these? Basically, we have very tiny skin biopsies, melanocytic tumors. And we get quite limited DNA. About half of our samples have less than the desirable amount of DNA input. And then on the right, I show more of a sort of some ranges. So less than 3 nanograms per microliter, so very quite limited DNA concentration in an extraction that goes to SpeedVac, and then just whatever's there gets put on the array, and about a little over half of those samples will work. Samples in the, below the desirable, but more than 3 nanograms per microliter, the middle range, they also have a high failure rate, but less so. And then again, about half of our samples yield the appropriate amount of DNA that has been recommended. So here's one more example. This is a 5-year-old boy, atypical spitzoid melanocytic neoplasm, spitzoid referring to like a histologic appearance. On the left lateral knee, we see losses on chromosome 6, 9, and 13. And when we look at chromosome 9, and it's not very obvious at all in this image, but when we zoom way in, we can see better that there is a very small deletion involving 31 markers on CDKN2A, which is a very key gene in the transition from, to malignancy for melanoma. And so suggesting there's a homozygous deletion of the gene, which would then, be fairly diagnostic of malignant potential. But again, this is very difficult to be sure of with the amount of noise and the limited size. And so basically what we did was we all looked at the case and consulted with each other, reviewed the findings, and we decided that this would be called a possible or questionable loss. And instead of saying for sure, and recommended following up with P16 stain, which would be the protein product of CDKN2A. And again, I also wanted to point out in this particular case, so we have a monosomy 9, and then on the other chromosome 9, we have a small deletion. And so potentially, if that is true, then we have homozygous loss of that region. But again, it's not completely certain. And so this creates problems for counting the number of CNVs, which is sort of our baseline for determining whether this might be on a genetic basis malignant. And so is this 3 alterations? Is it 5? So we have basically monosomy 9 is described in segments based on our nomenclature, just reporting. So if we count segments, we get one number. If we count true alterations, it's less intuitive, and I think that still is a bit of a challenge. There are some hallmark alterations in spitzoid lesions. Gain of 11p is one I mentioned. Disruptions of kinase genes. This one on the top right has a disruption of ROS1, breakpoint there. When unbalanced, then we can see it on the array, otherwise we would not. Homozygous deletion on CDKN2A, of course, would be very favoring malignancy. And loss of 3p is also commonly seen in BAP1 inactivated melanocytic tumors. These can have spitzoid morphology. One more case had a gain of 15, but it was quite subtle. We re-extracted. And this one had really, I think, 30% tumor. We really are try for 40% tumor on these samples because of the noise. And in this case, I think actually we were there, even though the pathologist impression was 30%. I think it probably was more like 40% based on my own evaluation. And so what we might actually have is just basically a population that has not yet reached the point at which it's the dominant clone in the tumor mass, and this can happen. And so basically what we're trying to find when we do the tumor array is the dominant clone, because that's kind of also a harbinger of malignancy, whereas I think early on there's multiple probably evolving sort of clones until it reaches malignancy. So not really sure at all, but again, this sort of highlights the need to have conservative thresholds for tumor content to make sure that we're adequately detecting abnormalities, and also to remember that we may occasionally have subclones. And so I have run out of time, but just wanted to highlight some of the areas that we could work on as a scientific community. One would be, of course, we needed blinded expert pathology consensus, perhaps from independent reviewers on the same data, which I'm hoping will happen in the future. We, of course, need disease outcomes, and that can be very laborious when patients are going back to their primary center, and these are simply consultation cases. We need to know if we have interlaboratory agreement and come up with standards to determine what really, when we count our CNVs, what we're counting and what we're not counting. And again, come up or formalize an algorithmic approach to using SNP array. And with that, I would like to thank my lab and the lab directors, Dr. Thorlin and Bond, and of course, all of my collaborators and the array lab technical specialists that I work with. Great team. Just feel lucky to be part of it. Thank you.